Assalamualaikum warahmatullahi wabarakatuh. Good morning everyone. We are from the group 2. We will presentation of epidemiology of plant disease. And before we start, let me introduce of our group member. The first is Natalia Fridolina Bau with the last three numbers is 058. The second is Zaidan Nuha with the last three numbers is 060. The third is I am Aldy Putra Pratama with the last three numbers 061. The fourth is Niranian with the last three number is 062. And the fifth is Chiara, Denise, Ante with the student numbers is Pil 210531, and the sixth is Parsley, Thyme, Hipolito with student numbers is Pil 210575. We will start with definition of epidemiology. Okay, the term epidemics come from the Greek, where epi means about or destroy and demos means the community or the group. Epidemiology can be defined as the study of about the epidemic and the factor that influence it. Epidemiology consists of a complex concept, definition, and analytical procedure. According to van der Plank's, epidemiology is the science of plant disease in a population. Nelson defined that epidemiology is the science that studies the factor that cause affect the rate of disease progression. Meanwhile, according to Kranz, epidemiology is the study of population dynamic but from numerical aspect, growth and regulation of plant and pathogen population. The most important thing in this case is that epidemiology is more focused on population phenomena.
The importance of epidemiology in agriculture. Understanding epidemiology has at least two goals, namely scientific or intellectual rules and practitioner rules. A science knowledge of epidemiology in charge of disease management technology. Both are important, where the first is understanding of behavior disease in time and space, while others which as first understanding to manage disease. It is understood that plant diseases are a part of nature which is impossible to dispel. Therefore, knowledge of them is very needed for the purpose of preventing an epidemic that could harmful. We need disease management to find out how much losses and how effective control strategies and reduce loss and protection. Epidemiology provides the basis for evaluation of control needs, control efficacy and stability control.
Disease progression. Disease on plants usually starts out at a low level, a small number of plants affected and a small amount of plant tissue affected. And it becomes of concern to us only when its incidence and severity increases with time. When we look at some examples of plant disease epidemics from the published literature, we not only notice that the incidence or severity starts near zero and then increases dramatically, but we also can discern some distinct patterns of development with time. In general, plant disease epidemics tend to be either roughly linear or exponential in the early stages, and they tend to level off as they approach some limit. Types of pathogens and epidemics. Number 1. Monocyclic pathogens and epidemics. Number 2. Polycyclic pathogens and epidemic. And number 3, polycyclic pathogens and epidemic.
The first is monocyclic pathogens and epidemic. Monocyclic pathogens complete are all of one pathogenesis cycle in one growing season and have one maximum generation per season. In general, there are three types of plant disease that tend to produce only one infection cycle per host cycle. The first is post-harvest disease. Not all post-harvest disease produce monocyclic epidemic, but in many cases, the infection that result in storage rats have either already occurred before harvest or occurred during the harvest and post-harvest handling before the product goes into storage. The root progresses during storage. A new inoculum may be continuously produced, but unless the storage product is handled again to disperse the inoculum and create new infection sites by making small wounds. There are no new infection cycle. Note that the pathogen is not inherently monocyclic but is concentrated by the environment to produce only a single cycle of infection. In other environments, the same pathogens might produce polycyclic epidemic. The second is disease caused by soil-borne plant pathogens. Many of the root rots, vascular wilts, and other diseases caused by soil-borne pathogens also produce only one infection cycle per crop cycle. The inoculum generally is some kind of survival structure resistant to desiccation of freezing, such as Sclerotia, Chlamydospores, or Ospores in the soil, or Mycelium in crop residues. The inoculum is dispersed in the soil by plowing and feeding the land and turning under the crop residues. As the roots of the newly planted crop grow through the soil, they encounter the propagates of the pathogens imbibed in the soil matrix and become infected. The epidemic progresses as new infection occur, but since any new inoculum that is produced is not dispersed until the soil is again cultivated, there is only one complete infection cycle per cropping cycle. To be sure, not only soil-borne plant pathogens produce monocyclic epidemic, and one must be very careful to understand the life cycle of each pathogen before drawing conclusions about its epidemiology. 3 is the mycyclic rash. Some rash produce no Yorkians per stage or repeating stage on a single host, and the inoculum product on the one host species generally must infect a different host species. The alternation of hosts appears to have evolved in adaptation to the annual growth cycle of the host, and we see one infection cycle on each host per year. An example is the Cheddar apple rash, where all the inoculum that infect apples comes from coals or red cheddars or junipers, and all the inoculum that infect red cheddar come from apple leaf and fruit. The epidemic on apples occurred during a 4 to 6 week period of basidiospore production in the spring. A second monocyclic epidemic on red cheddars occurred during a brief period of ichthyospore production in late summer.
So the next one is the polycyclic pathogens and epidemic. So this is the second type of pathogens and epidemics wherein there is more than one generation per growing season. So you have to take note of that. If the monocyclic pathogens and epidemics only have one maximum generation per season, the polycyclics have more than one. So an epidemic occurs when a complete and repeated infection begins. The infection is followed by the development of pathogens, the formation of new inoculum and new infections for a season. Some pathogens have many generations per year, so propagules are produced almost all the time according to the weather. So one example of this type of pathogen is P. infestans, which causes late blight on potatoes and can have several generations per season. By the way, blight is a plant disease that is caused by fungi. It's like a mold or a rot. So P. infestans survive season after season on diseased potato tubers left behind after harvest. The initial inoculum consists of the resulting sporangia on infected tubers spread by wind, and if perched on vulnerable tissue where there is free water and the temperature is right, it will germinate and host penetration occurs, resulting in a lesion. At an ideal temperature and humidity, the fungus will produce sporangia and infected tissue for 4 to 6 days.
Number 3. Polytic pathogens and epidemic. Monocyclic and polycyclic pathogens usually occur in one season. In some important pathogens, it is necessary to consider population dynamic and disease progression in season. If the pathogens can survive for a long period in a large population, then the inoculum at the end of the season is equivalent to the number of inoculum at the beginning of the season. If the inoculum increases from year by year, the development of the pathogens in a number of seasons exponentially regardless of its development in a season. It is called polytic pathogens. For example, Ceratocystis schulmi, or including Dutch elm disease, is monocyclic in a season, but Dutch elm disease increases exponentially when in several seasons. The fungus survived the winter on infective plant and died of elm. The elm beetle contaminated with its canidia when they land on this part can transfer it to the plant healthy and when they eat. Most infections occur in the late spring and early summer when the pathogens is ready to colonize fast-growing plant. The dynamic of the disease in several areas is exponential. Such dispersal mechanisms make it easier for pathogens to survive between crops or between seasons. The mycelium can survive in a living host. Some pathogens persist in the form of Canidia or chlamydospores in plant debris, others in the form of sclerotia on soil or plant debris. Other pathogens such as nematodes can survive in egg form, cysts in galls or in dormant form. Virus and bacteria in general survive by association with plant tissues. Next will be read by Adam.
Pathogen spreading mechanism. An epidemic occurs when a pathogen is able to spread to a sufficient area wide and have the opportunity to grow there. The spread of pathogen in this process of moving the pathogen or inoculum from the source to the site others. Spread of pathogens can occur actively or passive. Passive spread that plays a major role in causing disease, namely by wind, water, animal, especially insects and humans. Some pathogens can spread actively, for example nematodes, the spores, and motile bacteria. There are three types of inoculum are able to move in relatively short distance, or perhaps only few millimeters or centimeters, by using arm strength so that it allows effective to terms of disease progression.
Thank you so much for your attention, everyone. If you can have a question, you can type it in the forum at the Spada. Okay, thank you. Goodbye.